Good afternoon, everyone. Welcome to the CDSA Wednesday webinars. This is the last of our series on the digital solutions for clinical research in the pandemic world. We're going to speak about digital solutions to support trial conduct. I'm Usha Menon, and I'm professor at the MRC Clinical Trials Unit at University College London. But I'm here in my CDSA hat, where I'm strategy lead. We have two talks for today. First, Dr. Nitya Wadhwa will speak to us about platforms to support clinical trials. Dr. Nitya is a clinical trialist, a pediatrician, and she heads CDSA. She has really first-hand knowledge and about these platforms, and I'm sure you'll enjoy her overview. We will then go on to talk about biobanking, which is now increasingly critical in so many of the studies we do. And of course, COVID, there's been a lot of biobanking in COVID. And for that too, it's really important to have platforms that make sure the processes are secure and the samples are well stored and can be accessed. And Dr. Pallavi Kshetrapal will speak to us about digital platforms for biobanking. Dr. Pallavi is assistant professor at THSTI, and she's a senior scientist and leads the effort, the biobanking effort. So she supports all the teams at THSTI that have large cohort and other trials with their biobanking. So with no more words, I will hand over to Dr. Nitya, who will take you through platforms to support clinical trials. Nitya, over to you.
Thank you, Usha. I'll just share my, just give me a minute. Okay, just. Sorry. Okay. Yeah. Okay. Can you see my screen? Okay. Good afternoon, everyone. Today, we will be discussing the digital solutions to support the trial conduct phase in the clinical trial life cycle. You can see the span of the clinical trial life cycle over here, and it starts from the study design and grant application to planning and setup, and then comes the conduct. And this is, of course, followed by analysis, report writing, publication, dissemination, and all. The conduct phase itself has different aspects to it. There is the data acquisition, there is the data monitoring and safety monitoring, then there is the data management aspect. And today we will be talking about digital tools or solutions that can be used for all these different aspects of the conduct phase of the clinical trial. In the last webinar, so participant recruitment and retention is also an aspect of conduct. And in our last webinar, we did touch upon the technologies that could be used for recruitment, like e-consent and the different transmedia tools or visual formats that could be used to aid informed consent and make the understanding simpler and help in retention as well. Today, we'll touch upon the tools for data management, trial management, good documentation, and the monitoring aspect. Data management is the process of collection, management, and curation of data in compliance with best practices and regulatory standards to ensure that the data is complete, accurate, reliable, and processed correctly. Traditionally, paper-based case report forms were used in the past, where data was captured on these paper case report forms. And then in the 1990s, when the EDC system came into being, this data was then transcribed or entered into the EDC platform. But there were certain, this kind of data management platform was prone to some problems, like the systems were offline and so they were isolated. This was prone to data entry errors as data had to be transcribed. There were delay in timelines from data capture to data cleaning, and there were concerns regarding data security and backup. Then came the computer and web-based systems of electronic data capture, where here, I am not sure whether you can see my pointer, but you can see here the data is captured directly on a electronic interface, which could be a tablet, which could be a mobile. And from here, the data is transmitted into the server, which could be cloud-based. It could be a server at the data management center, and it also reaches the data management team as well as the monitoring team. There are several benefits of this electronic data capture. One is that there is quicker access to data, real-time access. There is less time spent on query management. There is quicker availability of the data for the analysis. There is more accuracy in the data that is captured because the EDC platform, there are options available to build in checks into the ECRF, which can prevent inaccurate and illogical entries from being made. Then some of the calculations can be automated. For example, if you have the date of birth, your age can be auto-calculated. If you have your last menstrual period date, your current gestational age at the time of pregnancy can be auto-calculated. Then because it's a computerized system, of course, the entries are legible. There is no chance of the entries being illegible. This is more cost effective, although initially the investment may seem to be very large, but in the long run, and especially if you have multiple projects that are happening and large multi-center trials being done across countries, across continents, this turns out to be cost effective. This is very organized, and so it increases efficiency because you have a user-friendly navigation, you have search options, and everything is stored in one place. Then, of course, the data is secure, it's password protected with a user account, which is role-based, and there is compliance with all the regulatory requirements. Another benefit is electronic interoperability. This means that these EDC systems, they use standards that are defined by CDISC. And if there are other systems, clinical systems that use similar standards defined by CDISC, then you can integrate the data. And this will make things easier. You don't have to enter the data. Like from the lab, you can straight away integrate the data into the system, and it makes systems more efficient. And of course, like I said, if you have multiple projects, large multi-center trials, and you're handling a huge amount of data, this is hugely beneficial. I'm just going to walk you through an EDC platform and how data is collected in this, just to give you a glimpse of the benefits of an EDC platform. So this was something that was developed in-house, but you can see the user is entering his unique ID and password, after which he can get access to the system. This is just the dashboard. And then we have the listing of the forms that are there. The user now goes on to start the data entry, where he enters the ID. The date and time is auto generated. And then he goes on to fill the date of birth. He makes an inaccurate entry and goes on to enter a date of birth which has not yet come in January of 2021, and this does not accept that date of birth. So he has to go back and correct the inaccurate date that he had mentioned. Then he goes on to enter the time of birth, and you can see the age is auto-generated. Then he goes on filling data in the form. He misses out on filling data in the severe chest and drawing variable, goes on to fill the axillary temperature, but he makes again an error in the units of the axillary temperature, and there's a pop-up to say that there is an inaccurate entry, which he goes on to correct. And then he continues with the data entry till he reaches the end of the form. And tries to save the form. But the system does not allow him to save this because he had missed out on one mandatory field, which was on severe chest and drawing. So he has to go back, complete that entry, and then he is allowed to save the data. So this is just to give you a glimpse. Okay. Then the electronic data capture system has other benefits. So what you can do is when you can create electronic CRFs, and then they can be saved in a library. And this is particularly beneficial if you have multiple projects, because you can then just take the CRFs out of that library and use it for multiple projects. So it eliminates the need to, recreate commonly used forms that are there. It also promotes data standards. So this is another benefit of the electronic data capture system. Then besides that, most of the commercially available electronic data capture systems have their data security in place. They are password protected. They have electronic signatures. They maintain a clear audit trail for traceability, where the complete history of record keeping, who created the record, who modified, who deleted it, who made it obsolete, along with the date and timestamp is recorded and stored. The system are validated. Then the data is in an encrypted form, especially during transmission and storage. There is secure data storage, and the data backup processes are in place. And as they are in place, then they comply to all the regulatory requirements, like the 21 CFR Part 11, and also the HIPAA requirement on Health Insurance and Portability Accountability Act. Okay. We move on to another technology that is now used for the clinical trial management. It's called the Clinical Trial Management System. It's a suite of e-clinical tools to effectively plan and manage your clinical study portfolio right from startup to the study close. It increases efficiencies because you can plan, you can track, you can monitor your study effectively, and there will be an oversight right from the startup phase to the end of the study. You can also perform budgeting within the trial management system if you actually exploit it to its fullest. Then study teams can work together within this trial management system, and there can be collaborations. Then the systems have automation where visit reports can be auto-generated. So once you've completed a visit, whatever observations you have, it is pulled out and compiled into a report, and that can be auto-generated, and it can also be emailed to the stakeholders, the concerned stakeholders. If there is a payment that is linked to a visit being made or to a subject or participant coming for a visit, then once that visit is complete, the payment can be generated. The payment mode can be generated, and payments can be made. And of course, the system, like the EDC, if it's commercially available, it is secure, it is reliable, and it is compliant with all the regulatory guidelines. The advantage of another advantage is that it can be integrated with other digital platforms like the electronic data capture platform, the trial master file module, creating efficiencies in the whole management of the trial. I'm just going to now take you through some of the aspects of a clinical trial management system. So one is, you can of course look at, like, if you want to review the schedule of participants for that day, you can access and you can look at whatever participants are scheduled to be coming to the site on a particular day. Another aspect is monitoring monitoring trackers, as we call them. And these are trackers which we have now customized and actually upgraded, so that whenever a monitor is monitoring the data or the informed consent or the site file, whatever observations the monitor is making, they can be logged in for each and every participant, and so that from this data, then you can come up, look at, you can do some statistical analysis, because you have a denominator. You can come up with trends that are there in the monitoring observations, what action has to be taken based on these trends. You can come up with the proportion of monitoring observations along with the confidence intervals. So I will just walk you through one of the trackers that we'll develop. This is the these are the trackers that most monitors would use. One is for informed consent, then for data monitoring, for protocol deviation, site file, and for the IP tracker. I'll just walk you through the ICD tracker. So here, so this is the tracker which we have actually developed, and you can see we've created drop-down menus for various fields. So one is the type of monitoring, and because it is informed consent, mostly it is done on-site. But in the pandemic situation, there could be situations where an off-site monitoring is done, and so we've kept the option of off-site monitoring for the ICD tracker. The screening ID, participant initials, if the CTMS is integrated to the electronic data capture system, then these IDs and the initials are pulled in directly from the EDC platform, and the monitor does not have to make an entry over here. So it makes things more efficient and faster for the monitor as well. Next comes the ICF version. So the default entry over here is the latest approved version, because that is the version that should be used. But in case there has been an error at the site and they have used an older version or an incorrect version, then the monitor has the option of selecting that from the menu. Then, of course, they'll categorize the observations that are there. This is a very exhaustive list and has been developed after a lot of brainstorming among the team members. And then he'll go on. The monitor will go on to categorize the classify the observation, whether it's a deviation from GCP, from protocol, from SOP, and then categorize it as minor, major, critical. And then after that, mention the corrective monitoring action that is required depending on the category of the observation and the deviation that is there. And so on. Will go on to complete the status of the observation, whether what was the mitigation plan, whether sensitization was required or retraining was required, and if there is any notification that has to go to the ethics committee or the sponsor, or to both, depending on the category of the observation that is there, and goes on to complete this. This is just to show some of the data that can be generated from these monitoring trackers that we have developed. We can look at trends in the monitoring observations, and they then can take decisions on the action required based on these trends. We can look at the percentage of the errors that are there along with the confidence intervals. Of course, most of the clinical trial management systems have inbuilt customized reports that are there. But if you want to customize a report that is not there in the library as such, you can go on to do some programming and customize and develop a report of your choice. We move on to another digital tool for trial management, the trial master file. The electronic trial master file, really. So conventionally, all the trial master file is managed by the sponsor or the designee. And if it is a large multi-center trial, then there is a humongous volume of documents that are there, and it's very difficult to organize and manage it and retrieve documents from the trial master file. So having an electronic trial master file will help store, organize, and manage the documents in a more organized manner. There'll be efficiency and quality. There could be instant document retrieval. You could pin your favorite files and folders. You could track correspondence, trial-related documents. You could even tag your documents and create a metadata card where you could name the document with who created it, with the date, the time of creation, and links could be created for related documents. There is an automated version controlling in the trial master file module. And just like any other module, there is a quality control workflow where the document has first to be created, then it has to be approved, it has to be reviewed, it has to be approved, and then it is finalized and signed off and uploaded. So far, we've been talking about the operational efficiencies and the timelines efficiencies that are created by using these digital technologies for the conduct of clinical trials. But there is strong evidence that there is an escalating cost for running these clinical trials. In fact, in 2013, it was seen that the average cost of a participant in the US for in a trial is around 38,000 US dollars, which is really humongous. And so a report was commissioned by the US Department of Health and Human Service to see if costs could be managed by tackling the inefficiencies that were seen in the operations. And so they analyzed some mitigation strategies, like they used electronic health records, if you could reduce the source data verification, if you could use mobile technologies or electronic data capture platforms, or you could use at-home testing or testing in a lower cost facility, or priority review. They looked at these mitigation strategies and to see if they could costs could be reduced. And it was seen that it was concluded that even when they used even a single mitigation strategy, like just home testing alone, they could reduce the costs in phase 1 of running of phase 1 trials by 16%, of phase 2 trials by 22%, and of phase 3 trials by 17%, which is really huge. So, and when you look at the impact on the net cost for each phase, this was really significant. So the leveraging of digital technologies in clinical trials not only will cause operational and timeline efficiencies, but also cost efficiencies. Another advantage is safety monitoring, because when you're harnessing the ability to collect the data continuously, you can improve the detection of infrequent events. Of course, one will have to make sure that the tools that are being used are reliable and validated. Another digital tool that can be used for trial management is source data verification. And really, this has, become a hot, tool in the pandemic area, in the pandemic era, when on-site monitoring had become very difficult. It was difficult for the monitors to go online. And so there were possibilities of doing source data verification remotely, and some of the studies adopted this tool for or remote monitoring, so as to say, with source data verification. Of course, the advantage is when on-site monitoring is not possible, but it is a lot of teamwork where both the teams have to be working together. So there has to be someone from the site and someone from the monitoring team who have to be working together, because and there has to be an infrastructure at the site, like there has to be a scanner of the site at the site, because the site team will have to first redact the document, take out any identifiers, then we'll have to scan the document, we'll have to upload it, and then this uploaded file is then reviewed by the monitoring team at this end for source data verification. But again, I would say that the guidance around this, at least in India, is not very clear. There on guidance on whether consent is required from the participants, whether there has to be there has to be guidance on whether you have to say that this is a true copy of the source data. The MHRA has issued some guidances on remote source data verification, but not we don't have such guidance in India, at least. So we are moving towards digitized clinical trials, but we have to be there has to be caution, because there has to be regulatory oversight, and we need more guidance on data privacy and security. But really, at least in India, but will the pandemic fast track the guidance documents? That is to be seen. With this, there is another important aspect of conduct, which is biospecimen collection and then its storage. And I will now request Dr. Shetarpal to talk about this. Thank you. So I'll have to stop sharing.
Hello, everybody. Good afternoon. It's Pallavi Shetrapal here from THSTI. cold here, so I'm going to talk about biobank, correct? Just wait a minute and I'll try to share. Here we are. Hope my slides are visible. And here I start. So we have a team here, Dr. Shinjini Bhatnagar, Dr. Pallavi Kshetrapal, myself, Dr. Shailja Sapori, Savita and Lavjeet. So this is a very small team. Don't think about it that this is a small team. We were really supported by many of our team members who I was not able to list here because as Nitya presented, it's a evolving process and it's something which is related to the clinical trials and the studies. So this is how our biobank looks at the THSTI, and I'll just talk about why biobanking is so important. So let us see, this came on the Time magazine in 2009. So there were the 10 ideas which were listed here, which said will be changing the world. And they were changing the world at 2009. Now, what happened at and this was the first biobank which the NCI was establishing in the US. There were other biobanks which were already established in UK, Sweden, Norway. So now, what happened in 2019 suddenly? Again, it came on the Forbes magazine that yes, biobanking is still changing the world. Now, what was the new thing here in 2019? And the new thing was that it is all artificial intelligence driven processes, data analytics and robotics. Rightly mentioned by Nitya, everything was like you can keep a register or you can keep an Excel file. And now you have all these data analytic platforms or digitalized platforms, as we call it, which will help us not only to keep the records in a way which we can retrieve very fast, but also are cost efficient. That was one thing which she also pointed out. So let us go ahead with the topic and the scope of the talk today. What are biobanks? Why are the biobanks necessary? And how does one establish a biobank? I'm sure these are the questions which will be arising in your mind now. Now for biobank, I'll go very, very deep in a preliminary basic thing, which I'm sure everybody would have touched upon. Whenever we used to get pocket money, that's the money which we can spend out here. Father and mother used to say, okay, you can spend it whatever your needs are now, but try to bank it or keep it aside so that when after 10 years, maybe after 10 days, in that childhood, you will change your objectives or you will change what you need. So that bank of money will help you. Now, not differing from the topic of the biobank is a collection of human specimens. It could be a collection of plants. It could be a collection of microbes, which are being collected in prospective cohorts, maybe a trial, maybe observational study, but for their primary objectives. The best is to bank whatever is left with proper compliance and rules and regulations so that after 10 years, when you have new this is generated, which you have, will be, you will be able to utilize leftover specimens or maybe something which you are biobanking now. Coming to the next point, that how does one see that really, like what talked about in the Forbes is really important? That is evident by the number of publications over time. So I hope you're able to see my pointer. In 1996, there was one publication. Now you come down to the point 2009, where they established that, okay, we'll have a first biobank, and it came onto the Time magazine. There were 83 publications. Now you just see the jump here from 2009 to 2019. And this was don't worry, this is something where this publication came out. So that is where we are stuck here. This is not December yet. But yes, you can see the trend that these publications have increased over time, which talk about biobanking is a field which is very important for societal benefit. Now, the interesting thing was what to biobank. So what people started with was disease-oriented biobanks, which is very disease-centric. And yes, cancer was one of them at that particular time and still is. So now cancer biobanks were the ones which came up. So the most of the publications we see from that era are the cancer biobanks. Then what happened? The second thing is consenting. Now, what are the consents one needs to change when you're biobanking specimens? You have to be very clear that you will be using such specimens later on, that is retrospective of the study. Then came the concern of ethics. We need to know that biobanking something, we cannot be selling off samples. We need to have a process. So that is where I was involved, and ethics committees really had to move up what could be bio, what could not be bio, how it could be utilized. Then afterwards, after all these processes were set, now biobanks were full of biospecimens, so people understand what are the biomarkers for different diseases. Prediction medicine started. It became the era of bioethics, then public health. The least publications we have are the personalized medicine. Now, this is where most of the research is now going to happen. And what they need is all these biobank samples which have been biobank. And I have also kept some of these references here if somebody is needing here, so that they can just pull out these references and read it a little bit more. Now, just coming to the personalized medicine, everybody talks about what is personalized medicine. This is a dream, but now it's coming true. And if you see on the left-hand side of the panel, there's one green tablet which is being given to everybody, and it depends. Some of the people will react to it, they'll get better. If you see the effect size here, some will have no effect for that medicine, and some might have an adverse effect, which will be, yes, definitely lower. Now, with the help of biobank, what can happen is, because here is the place where the biobank is, where you're storing the blood, you're storing the DNA of the particular person, the RNA, the urine, and the tissues analysis or the tissue specimens. So what happens when you have a drug? Instead of giving the drug completely to the person, it's advisable. This is something which is again not in total practice now, but some people have started working on this, that you can test the dose on the specimens which have been collected from the patients or the participants. And then you can have a personalized medicine depending on the DNA structure of the person, or maybe how the person is reacting to a particular drug, at least along with their tissue or the DNA. And this really says that it's going to be proving much effective, and you can see the effect here that yes, personalized medicine would be better. And as I said, studies are still going on. That is why we have lower publications here, and many people are doing it. Now, what is a biobank? I have talked about why it is necessary. Now, what are the processes that go inside a biobank? Are very simple, but everything has to be in a guideline manner. So we also started very slow. So we knew we have to collect something, we have to process something, we have to store it, and then we have to distribute it. Distribute it to the community, to the research community, to the academic community, so that some answers could be sought in the biology field. Now, if I talk about collection, the first thing which I mentioned is the ethics. So these are blocks which have to be really answered well. Why? Because these concerns which come from the ethical committee are the concerns which are being arisen in the participant also who is participating in the trial for biobanking. What are they going to do with my sample? Why do they need my sample? Is it ethically safe that I should give my sample to them? Whether they will keep my records properly? So these are things which come up, and that is what, when we look for any biobanking proposal or where the samples are being biobanked, we have to go to ethics committee to sort all these answers for. And once it's cleared, we will be able to clear that much better when a participant is asking such questions. And then we collect the sample. Next comes processing. Now, we have already told the participant or the patient that we will not be keeping all his records. Everything will be anonymized. So the best way to anonymize is barcoded. So what we do is, we thought, okay, fine, let's barcode it. We understand what this particular sample will be. And then we need to make visual SOPs so that people understand that all these specimens which have been collected are precious enough and need to follow a particular SOP which is developed in a way which can be implemented. And then these SOPs are implemented into different studies to process these samples to a place where they can come for storage at the biorepository. Now, the interesting thing is the distribution. How do you distribute the sample? Somebody comes into you, you want to get, I want this sample. You can't give it to the person. So what you need is a governance process, which is an access control committee. You can have access to the specimens, but you need to know whether it is scientifically feasible, what you're talking about, what is the scientific merit of your question. So these are the things. And the second thing which a biobank needs to know is that all these samples which need to be distributed have to follow the ITA regulations. And subsequently, I'll get to the talk that what these ITA regulations and how you can follow them. Now, we also started very slow, as I said, and we just knew these four things, which is collect, process, store, and distribute. And we started implementing our SOPs and made the participant form. We made a participant form in a way, also got ethical clearance from the ethical societies of the different hospitals or the study sites. Then we went into data collection, which was very briefed nicely. The sample collection, which is the maternal sample we were generally collecting, the sample quality had to be evaluated, and I'm going to come in a bit with that. And then comes the biobank over here. So these are all the processes which have to go hand in hand. Biobank on itself is nothing. It has to have a process. Now, the first thing was that in the biobank, when you need to store the samples, as I said, the lab processing SOPs were first tried out at a lab setting and then were implemented into the study. Second was the infrastructure establishment, not only for the biobank, but also from the place or the study site where these samples are being processed. So if you don't have something, when you make a recipe, if you don't have the right type of ingredients, you never have a finished product. The same way, we needed to know that the infrastructure, not only at the site where we are storing the samples, but also at the site where the samples are being processed, has to be in place. Third was the SOPs which were developed for the biorepository, where the samples go, how the samples come in, how do we receive them, where do we store them, because we are not talking about 1 or 2 samples, we are talking about 1 million samples out here. Now, the fourth step is well-defined authorship policy. This comes into being when you are collaborators on the project or your collaborators on a specific program where biobanking is also being done. The fifth is the governance and the data sharing policy, and I'm going to talk about it, how we try to maintain this governance and data sharing policies on all the specimens which have already been and the data which has already been collected. A very brief about this cycle, and I'll take some time here. So if you could bear with me. So one is the barcoding, where you can decode the samples. You have no personalized information about the participant. So you barcode it. Once the barcode printing is done, the scanning of the biospecimen is done at the study site. Then the temporary storage, because your study sites are not going to be where you're going to be storing your samples. So you have to think about that too. So you have liquid nitrogen shippers, which are known as dry shippers, which can maintain the integrity of the sample at lower temperatures and be very useful while you're transporting your samples. It comes into the THSTI biorepository. The important thing here is, because all these samples could be infectious. So whenever it comes into the biorepository, you need to have some sanitization protocols. The second is that it comes into the biorepository. It has started from here, but it has taken a long journey. So you need to know that your sample, which has been collected from a participant, has been well received at the biorepository. There could be some more processing which is necessary. And so we also have built up a lab in THSTI where you will need some specimen post-processing also. So you have to think about this also, that biobanking can be done with some samples being pre-processed. Some you will have to process a little bit more if you want to store them for a long-term storage. Then you need to aliquot the samples. You can't keep all the samples in one tube, correct? As we say, don't keep all your eggs in one basket. So you need to keep them in different freezers and in different aliquots. So at least if there's a mishap of something where you'll have contingency plans, mind me, but you should be able to retrieve some of your samples. And then finally, the storage of the biospecimens. It's just keeping something in the freezer. Is it so simple? No. Let me tell you why. Because you need to abide or have the compliance of a few biobanking agencies, which I have mentioned here, and the best practices that you need to know that how do you do it. So let me tell you, we made our SOPs and then we had to revise our SOPs using all the guidelines we could learn from them. Some of them are open source, some of them you will have to get registered. So ISBR, which is an International Society for Biological and Environmental Repositories, you will have to register yourself as a member. Thankfully, we are members. So we get first-hand information in the biobanking field. What are the best practices if they have changed any? Now, this is how we revised our SOPs. Let me tell you, this is a very busy slide, but very important slide, where you can maintain the quality management systems for it. You need to have certifications for the biorepository. Not all biorepositories need certification. It depends whether you really need to be accredited, because if you're going to talk about opening a biorepository or having a biorepository with your collaborators, or maybe you want to harmonize it with the other global biorepositories, then yes, your accreditations and certifications are a must. But there are some tools which you can utilize for opening a biorepository or maintaining a biorepository without having accreditations or certifications. So these are known as SPREC standards, standard pre-analytical code. What do you mean by this? As I told you, when you make a recipe, you need to know what are your ingredients. So the sample can go bad not only by storing, but the sample can also go bad at the time of collection or at the time of processing. So all that needs to be recorded and timestamped. So what we did was, while implementing our SOPs and rejuvenating them, we trained our personnel, which we were already doing. So you have to have GCLP training, and you also can go for an ISO 9001-215 and an internal auditing training, because this will help you not only what is going bad at the collection or the processing, but also at the storage. So there would be internal auditors who are well thought enough or well read enough, because they have undergone a training, so that you can look for those critical risk factors which could go bad, and basically your sample integrity would be affected. So that would be a very good feedback from your own people you will be getting, and then you can use the kappa methods. Second was the management operation. So yes, as I said, you need to have a monitoring facility, because your freezers where you're going to store your samples could be -80°C, or you could also store them below -80, that is 196 liquid nitrogen. But what all processes you need is that you will be able to monitor it every minute, because they are precious samples which are from a study and from a participant who has abided by you, who has confined in you. So that is why you need to also have the FDR 21 CFR Part 2 compliant processes to maintain the biorepository. Now, these are again digital platforms. So that is the theme of the talk. So here again, you have quality management, where you need to know your data delegation logs, the duty delegation logs, the training logs, the inventory, the temperature log. If there was some incident or accident which has happened, what was the lower temperature, what was the higher temperature, and this compliant software is definitely going to help you with that. Real-time tracking and query resolution, because you have your built-up team, so they will help you with the real-time tracking and the query resolution. And if any, after all these contingencies you fail, you need to have your protocol deviation logs ready. If any protocol was deviated at the time of collection or the storage or processing, that needs to be recorded, and it should be recorded with all the data or the clinical data also, so that we know that this particular sample has lost its sample integrity and cannot be used for some specific objectives. Now, governance, we talk about this. Yes, as I mentioned, we have access control committee. That is very important, because you are just the custodians of the sample. You need to have an expert committee to guide you whether these samples can be retrieved for certain scientific experiments which would have some societal benefits. So you need to have your expert committees ready. And as I told you, you really need to have certifications or recognizations and different international societies which will help you time to time. And as I mentioned, biorepository is standing here, but it has its 4 pillars. One is the clinical team, where you get your samples from, because they abide by and they have their clinical trials or they have their observational trials. Second is the data management teams. Again, you have digital platforms for it. You have electronic data capture mentioned just now. And all this data is stored in servers. The same thing with the biorepository team, as I mentioned, the process of it. It will again come down to a server. And once all this is in place, then only the samples can really go for any research and diagnostic questions. Now, coming to the digital solutions, which I can just brief you about, which we have read and which we have really looked into also for digitalization, not only from the collection, but also to the storage, are the biorepository, the freezer works. And these are some of them. There would be many, but yes, all of them, what they do, I'm going to talk about in my next slides. What do they do? Now, if I need a specimen right now, because it has been already approved by the Access Control Committee, and I have around, say, 30 freezers, and I need one specimen, one freezer is maintaining 60,000, how do I go to it? So I cannot use my Excel sheets, or I cannot use my registers, correct? Wahi khata, that was what the word was in previous times. So what we do is we scan the code. As I mentioned to you that every participant has its own code. Every participant with the sample type has a very unique code. So these are UICs, unique identification codes. They can be either 1D here or they can be a 2D code. So once this is scanned, each specimen, each ELICOT will have its own unique code. And with this code, we can make a blueprint of a freezer, what type of box contains what type of specimens. And all this data has to be very well maintained, and very rightly said that whatever data is being scanned everywhere has to come to the servers. So we maintain all the data in the server so that we have backups. And these are the different types of reports one can generate from the scanning of a biospecimen or a unique specimen, that what it is, when was it collected, where has it gone. So all these things are there mentioned, and I'll just tell you with simple, you can say real-time examples from our study. So real-time monitoring of samples for pre-analytic QC using digital solutions. This is the code, 1D, 2D, what happens? So if you need to know what type of sample was collected, so this is the urine sample. Now, just one thing I forgot here, that these are in-house built digital solutions. You can also have them customized by different companies, which I just mentioned to you before about FreezerWorks and Open Specimen. So this was something which we had built up using our data management team and their expertise for time stamping, and also for the complete process till the storage. So this specimen is a urine specimen. This is the timestamp, when it was collected, what happens next, when was the centrifugation of this specimen which started, and when was it that it ended. So we have this particular set of parameters which we have put. Now, if it overruns, it will give a beep to us also, sitting remotely at a place, knowing that, okay, one of your specimens has overrun in a centrifugation step. So that tells you that we have an audit trail for each and every specimen which is going and being collected. Why? Because every unique specimen has its unique identification code, and everybody has the SOPs with them. So we know that there are some protocol deviations which have happened with the SOP. So once it comes to the biorepository, again, as I told you, so we also have the storage time. So we know exactly if we have 4 hours duration of a particular sample which it has to reach, and it has reached in its 4 hours, the light will be green. So that means that specimen has maintained its integrity. Now, this is the timeline how we begin with. So I have just gone about my talk here like a story, that how from a lab refrigerator where we were storing samples, we moved into storing them in the -80, so single or 2 -80s. And then we had a fleet of -80s, an arsenal here. But that meant following all the rules and regulations. And that is what I was trying to brief you in my talk, that these are the different guidelines that you people can follow to open up a biorepository and maintain it. So what happened was, from an area of 552, we went to 2,766. So this is our timeline from 15 to 20, that is 5 years. And from 5 deep freezers, we increased it to 23. Then we were only going ahead with NABL accreditation. Now we know that we need to have IQ, OQ, and PQ. This is installation quality, operational quality, and processing quality. When you install a particular equipment, it is the best. But you need to have the operational and the processing quality maintained for the following years, because this might decline. So you need to know that whatever equipment you're using in the biorepository is indeed the best. From 1 clinical study, we went into 5 clinical studies, and the number of biospecimens was from 1 lakh to 10 lakh. So this is our journey, and this is the complete Garbhini cohort, which is a pregnancy cohort which is going on in a district hospital. So these were the number of participants that we have enrolled in the study. These are the different types of specimens. I'm just going to name a few here, which is the umbilical cord, maternal saliva, the placental membrane, citrate, plasma, blood. So all the SOPs were developed for all different 10 or 20 types of specimens which are being collected. These are the number of specimens that have already been retrieved. Now, let me tell you one thing. The best biobank is an empty biobank. But again, everybody tells that keep some in reserve. What do you mean by that? So now, as I told you, biobank has the, you can say not even authority, it's the responsibility of the biobank to distribute or share these samples with the people who really, or the scientists or the industries or the companies who really would come up for a societal benefit. But then in that case, you should always try to keep some sample for, okay, sorry, you should also try to keep some sample in reserve, because today the need is COVID. Next, in 5 years, there could be some more challenging things coming up. So it's better to keep samples for next questions which are being developed also in your own study. So what happens with that is you have a 1 ml of sample. Somebody wanted 500 microliters of the sample. What you do is, this is your parent cell. So you see this, then it is the daughter cells. So these are the two daughter cells over here, which is the pink and the green. So you keep 500 microliters for yourself and give 500 to be shared. So this is how you can maintain your biobank also. I'm very pleased.
We're just sort of running out of time, so
yeah, last slide, then I can come up to. Yeah,
sure.
Thank you so much, Usha. So this is again which the biobank was repurposed for COVID-19. And the other thing about biobank is you should always have visibility and also transparency. So you could put it up on the Twitter, you could put it up on the Facebook, so that the participants also know what is going on with their samples. And the last is, yes, the participant, the engagement that we do with the participant in the study, at least with the COVID, is that when they enroll in our study, we have a thank you card for them. We have a small gift which will help them keep themselves sanitized here. And this is how we participate in the study. And the last take-home message slide is that what is the national preparedness? Yes, definitely we need to promote human health and well-being of the community, but then funding are the issues. You need a proper infrastructure to open a biobank, and also you need to do it with the guidelines which are already present. There are complex challenges, which is ethics, governance, intellectual property, and collaboration, which I just talked about. And what next is the virtual biobanks. The biobanks would be there, yes. All the information will be password protected. It will be on a cloud. And you can do federated learning, which is at one place you keep all your data and everybody can have access to it through its federated learning laws. Thank you so much. I would like to acknowledge BBT and THSTI and definitely CDSA for making me do this workshop. Thank you so much.
Thank you, Pallavi. I think Neha Chawla will be kind of chairing the Q&A, but we are sort of very short of time. So perhaps, Neha, we might only be able to take a question from each, from your list that is running in the chat box.
Sure. Thank you once again, everyone. So we've received a couple of questions from the participants while the speakers were presenting. So my first question is for Dr. Nitya Wadhwa. Dr. Nitya, are there any demo links available which will help navigate EDC, CTMS, or electronic trial master file, or do they all have to be commercially bought? So this is one of the questions we received while the talk was going on.
Okay, so I'm okay. Demo links for all these. So if you want to fully understand, if you want to fully understand how a process or a system works, you will have to. You can always, request for a demo from the vendor for the commercial application. That will help you explore and understand the full application in toto.
Okay.
Yeah.
Okay.
The other thing, I guess, is, I'm sorry, I seem to have some background noise. You could always ask one of the teams that is using it,
Yeah, sure.
one of your academic collaborators, if they could just show you. And most of us would be very willing to show you at least the system we have. It won't be like, so nearly every team is now using an EDC. So if you know a clinical trialist or a study team, they could share it with you. But like Dr. Nitya said, you can just feel confident and ask a vendor, and then you could have a good look at the system.
Right. Professor Menon, I'd like to switch to you now. How do institutes generally go about setting up a biobank? If you could share with your experience, please.
So I guess Dr. Pallavi has really gone through setting up the biobank as well as the digital solutions used. I think they're cutting down some tree in the background, so I'm really sorry for the sound. But with regards to the challenges, I suppose the first thing is to get buy-in from the senior team, from the leadership team at the institution, because as Alavi explained, it's not a small thing. Of course, for our own study, you have a small biobank for a small study. That is different. But if you have a large study or you're writing a grant for a multi-center study, you're going to house a biobank in your own institution. It's a long-term project, and it really needs real buy-in from the leadership. So if you got that buy-in, that's the really first step to then getting the budget, the infrastructure. And some challenges you run into, like in a hospital setting or an institute, is quite often space. When there is a commitment from everybody, you might still find, you don't have, as the pipeline grows, there is a space issue. But on the whole, it's all about of also finding somebody like Dr. Pallavi who's totally dedicated to kind of setting up that biobank and running it. Because, it's not, she too is a scientist. So many of the people who lead biobanks, they have their own research. But this research, this whole time they spend setting up your biobank, setting up the biobank, it's not something that really personally benefits them. They don't get a publication with their name or like it's really scientifically exciting because they were, of course, they have some projects which are running off the biobank, but a lot of work is to support other projects other than the core one that they are working on. So these are some issues that you need to think about.
Right. Thank you. Dr. Pallavi, moving on to you. What ethical principles would one consider or generally apply? I know you touched on it briefly during your talk, while obtaining and storing samples.
Thank you, Neha. So yes, I did touch upon them. And the first is, I guess, is the informed consent. It should be very crystal clear and transparent to the participant that he is being invited to a study where the samples are going to be stored and being used retrospectively, correct? And the biobank will only be a custodian. We are not the real owners. We are just going to be having it in custody, and the rightful use will be decided by a governance committee, and one of the participant or the legal representative is always there in that. Then the other thing, it has to be very clear that they can retrieve or they can withdraw the consent anytime during the study or after the study that they don't want their samples to be utilized. Yeah. So that is one thing. And the consent has to be not a very blanket consent also that you can use a specimen in any different type of study or any specimen anywhere. It has to be something which has to be well thought of. So that is the first ethical thing which has to be kept in mind. That's it. So any more?
I think we have time for one more question. So, Professor Menon, if I could switch to you, please. Are there any other issues with regards to data safety and security while using digital solutions in clinical trials or research?
Well, there's just one thing I want to bring up because you saw that Dr. Nitya showed her own bespoke solution. So, you have built a very nice system to bank the specimens. But the important thing is, and you've got all the data, you've got everything that is required, but it is about where the data is stored on your server. So you've got servers, you store the data. But it's really important to think about mirroring the data. Do you have the data held on another server if your primary server went down? Do you have a clear disaster recovery plan? And so all those security issues, one way about going, not having to think about that is, of course, buying a solution where in the cloud they are saving the data. And of course, they follow all the guidelines, which both the Indian government guidelines and the international guidelines, if it's a commercial solution. But if it's your own homegrown solution, it's really very much about making sure that you don't lose the data. So, because if you do a very good job, and it's a huge investment with regards to both the funding agency, the team, and the patients or community that have donated the sample. So it's all about having a disaster recovery plan if you have a disaster. And some people would have you may have everything in the institute, but if you're really concerned that, let's say, there's a fire in the institute and the institute burns down or something like that, you might even have the whole data then held at another site as well, physically at another site. So these are kind of things that you need to think beyond your own, wonderful data or commercial solution.
Well, thank you, everyone. Thank you once again, Dr. Nitya, Dr. Pallavi, and Professor Menon for joining us today. I hope the session was useful. Due to the paucity of time, we've only been able to cover some questions, but if you feel there are any unanswered questions or you'd like to interact more with the speakers, please write to us and we'll try and get them answered from the speakers for you. Thank you all once again, especially the three panelists. Thank you.
Thank you.
Thank you.
Thank you, everyone. And we hope to see you next year. Happy New Year.
Thank you.
Thank you.